everyone. Welcome to the PAPERI Bright Minds series, organized by the Pan-African PGS Education and Research Initiative. My name is Abigail Nduzo from Zimbabwe, and I'll be your moderator for today's session. The Bright Minds series highlights leading scientists whose work is advancing genetics, genomics, and biomedical research globally, while inspiring the next generation of researchers and innovators. Today, we are truly honored to welcome Dr. Jennifer Troyer. Dr. Troyer has more than a decade of leadership in genomics research administration and global health initiatives. In her current role, Dr. Troyer leads Genetic Alliance's global genomics strategy, strengthening partnerships across sectors, and expanding initiatives that connect genomic research with patient communities. Prior to that, she served as director of the Division of Extramural Operations at the National Human Genome Research Institute, where she led efforts to coordinate research funding, policies, and programs supporting genomic science worldwide. Throughout her career, Dr. Troyer has led and managed complex international research initiatives. At NHGRI, she oversaw grant portfolios focused on comparative genomics and genome function, and played key roles in major global programs such as the Human Heredity and Health in Africa initiative, which advances genomic research and infrastructure across the African continent. At the National Cancer Institute, she supported a sequencing technology development program and conducted research on viral host interactions. Dr. Troyer earned her PhD in genetics from the University of Connecticut and her BA in biology from Elam College. She completed postdoc training at the National Cancer Institute and Colorado State University. Dr. Troyer, welcome to the PAPERI Bright Minds series. It is a great pleasure to have you with us today.
Thank you so much. It's an honor to be here.
Right. To begin with, would love to start with your personal journey. You have a uniquely interdisciplinary background, spanning evolutionary biology, virology, immunology, and genetic epidemiology. What initially drew you to genomics, and how has your journey evolved over time?
That's a great question. Thank you. Yeah, I've always been interested in a lot of different things. And so I think that people looking at my career might think that it's sort of all over the place, but I feel like there's a very logical thread and logical connection between all the parts of it. Growing up as a child, I was always I loved animals. I was interested in the natural world. I spent most of my time outside. I was one of those weird kids that could spend hours watching ants or butterflies or, what they were doing, how they were working. I was endlessly curious about how things worked. And then in school, when my father actually said he knew that I would be a scientist long before I did, because in 5th grade, I came home and couldn't stop talking about how viruses were the coolest thing in the world. And and then later, I always really liked genetics at the time, learning about Mendel and crosses, and it was all very something you could figure out. It was like a puzzle, and unraveling that puzzle and figuring out, with genetics, with the 4 letters that are this code that encodes for all life, is amazing. And so, that sort of tied in with the other things which I like to do, which was solve puzzles and math problems and things like that. So it all sort of was very much part of my life and what I loved to go towards biology and towards genetics. That being said, I didn't know I wanted to do scientific research, let alone research administration. I didn't even know as a kid that those jobs existed, right? So I got to college and sort of knew that I wanted to do something science-y, but didn't know what. I took a lot of biology and molecular biology and chemistry classes. And where I went to school, that kind of put you on the track to be a pre-medical student. So that's sort of what I thought I was going to be doing, is going to medical school. And then I think, lucky for me, because I really am happy with where I landed, I did not get into medical school and had to sort of rethink what I was going to do. And then so I went back to school to get a PhD in genetics. And at the time, I was still thinking I would end up in the medical realm, which it's funny because this has kind of come full circle. Right now, I work with a lot of genetic counselors, but I thought maybe I would do genetic counseling. And then I started getting into the lab, and that research bug just kind of bit me. And at the time, we were doing fruit fly genomics, which, the fruit flies themselves are not all that interesting, but the fact that you could figure out stuff about what was going on inside their cells and their genes, and it was sort of in the very early stages of genome sequencing. And so my thesis project was trying to figure out how, using Sanger sequencing at the time, you could sequence through these highly repetitive regions, the heterochromatin and the centromeric and telomeric regions that just had lots of the same sequence over and over again. And, we had no idea what was in there. And now we're starting to figure that out. I think back on my thesis, everything I spent all those years doing now could be done in probably less than a week, with the way in which sequencing technology has advanced. But at the time, we were still trying to figure out how to do it. And so sort of, I think, halfway through my thesis, and I imagine most people go through this, I sort of had this existential crisis of what am I doing and why am I doing this and what am I going to do next? And I started looking around to see what people were doing with genetics at the time. And I've always, as I said, been passionate about the natural world and animals, and I care about, conserving the planet and conserving species. And so there was this group of people that were starting to use genomics to think about how do we save species that are becoming endangered and that are going through bottlenecks in their populations. And some of that work was actually going on at the National Cancer Institute, in combination with a lot of other stuff, and we can talk later about why that went together. So I went to a 2-week course on, it was called Recent Advances in Conservation Genetics. It actually still is happening. I highly recommend it. It was a great course for me. It was a life-changing experience, and met a bunch of people who were working on species that I cared about. And one of those groups was Stephen O'Brien's lab at the National Cancer Institute, who are the ones that were actually running this course. And so over the next year or so, with conversations with them, that's where I decided to do my postdoc and work. And it was very exciting work. They were doing a lot of different work with a lot of different species, and the project that I came in on was looking at the way in which feline immunodeficiency viruses affect lion populations in Africa. So it sort of combined all these things that I had always loved. I had always watched, the programs in my youth, about the lions in Africa, and so and I love cats of all kinds. And then it was, what viruses are infecting these lions and what are those viruses doing to them? And as I said, viruses I have always thought were just biologically very cool, very interesting. And so it pulled that all together. And so that is kind of what brought me into this whole area of the ecology and evolution of infectious diseases, which I worked on for many years. and sort of went from what are lentiviruses doing in these feline species, and how do they jump between species, how does those species transfers occur, and how do the species, what are the protections? So I got into immunogenetics and sort of what are the protective measures that are encoded by our genes and are allowed to evolve more rapidly than the rest of our genome so that we can fight against these viruses that evolve so rapidly. So that whole evolution piece really fit into also health, like what is happening to your body when it becomes infected. And so that brought me back into the world of human health, because at the time there were several, lots of studies being done as we started to do whole genome association studies with susceptibility and resistance to HIV. So I was involved in a lot of those genetic association studies, and trying to understand not just individual genes, but the whole genome impact on whether someone will become infected, will have a long latency or not, all those questions about AIDS and HIV. And so that was sort of my postdoc, and then beyond postdoc, I became a staff scientist at the National Cancer Institute, looking at these associations. And at the same time, continuing to do the work in other animals, because what could we learn from them, right? Like what happens in our species, but also what's happening in other species with similar viruses, and can we learn anything from that? And then my lab chief at the National Cancer Institute was retiring, and two things can happen to a lab when the lab chief is retired. Sometimes somebody can take over, and sometimes the government decides to close down the lab. So they had decided to close down that lab. But this was sort of at the point when whole genome sequencing was really coming into play, and Illumina and PacBio and Oxford Nanopore at the time, and all of those technologies where the ion torrent was in its infancy. And so there was a sequencing service lab at the National Cancer Institute that worked with many, all the PIs, the investigators across the National Cancer Institute, to develop genomic technologies and genomic tools to help them in their research. And so I became the director of technology development for that group to really work with the sequencing. How do we use these new sequencing technologies? How do we understand the data that is coming off of them? How do we build the bioinformatic tools to answer the questions that the investigators were asking? And so again, because I sort of had more expertise in virology, and we were working with HPV and some of the other, the cancer-causing viruses for the most part was a lot of the work that I did there. And so because I was at that time really involved in the technology, yes, but even more so the bioinformatics. How do we understand the data that is coming off of these new machines that are giving us this new type of sequence data? And especially a lot with PacBio, we actually had one of the first 10 PacBio sequencers. And so this like long read data was new to everybody at that point. I then ended up applying for a position at the Human Genome Research Institute to be the bioinformatics lead for their extramural program. And I think not everybody knows what the extramural program is. I certainly didn't at the time. So the extramural program is actually more than 80% of the NIH budget goes to funding research projects across the United States, but also around the world. And scientists work within that program to make sure that they're thinking ahead to what's going to be the next big thing, what's the next big problem we have to solve, how do we solve it, what kind of research needs to be done in order to answer these questions. So how should we be distributing these funds across all the projects, all of which are really good science, but how do we select which science we're going to support right now? And so I had applied for this bioinformatics job, and it just turned out like I feel like I've been very lucky every step of the way. First of all, I just want to say, like, all the work that I've done, and part of what I love about genomics is it's extremely collaborative. You're never doing anything alone. You're always working with this amazing group of people. And so I met this amazing group of people while I was interviewing that were working on this program called Human Heredity and Health in Africa. And it was, again, sort of in this world of combining all the things that I'm interested in and passionate about. I was just so lucky that on that interview, I met people that were working on this project that I had never heard of and didn't know was happening. But the whole goal of it was to increase the capacity for genomics research in Africa, on the African continent, to understand disease and health in African populations. And so it was both infectious diseases, but also non-communicable diseases. It was people, it was helping scientists. It was, so all of that was just really exciting to me. And it was popular, genomics in a place where there's so much information and so much variation in genomes that clearly was missing from our understanding of human biology and genomes. And so that was just really exciting. And it turned out that they were going to be hiring somebody. And so I was able to come and work on that project. And then the scientist who is leading it, Jane Peterson from the NIH side, and again, this was a huge collaboration with all the scientists in Africa, but also the Wellcome Trust and other people around the world. So from the NIH side, Jane Peterson was the lead of that project, and she retired, and I was there. And so I was able to pick up and move it forward, which was, again, I was so, it was just great timing to be able to do that. And when I joined, kind of the seeds were planted, but it hadn't started to grow yet. So I got to watch it grow and work with it. So that's we're almost done. But see, there's a logical progression from step to step. And then, because that was a Common Fund program, and Common Fund is the director's program, and they're supposed to be large, transformative, high risk, but high reward, and they are time limited. They only last 10 years. Okay. So H3 Africa, not as an entity, they're still going, they're still working, they're still doing great science, but as an NIH funded program was ending. And so I was looking for what's the next thing that I'm going to be doing. And our director of extramural operations was going to be retiring. And that position is kind of different in that it sort of sits at the hub of everything that's being done in that grant making program. So it's grants, it's the financial management, it's the scientific peer review, it's the policy oversight, it's the long-term planning and strategic planning. So it just sort of holds all the pieces together, and it helps everybody that was kind of doing the job that I was doing. It helps them do their job better. So it was kind of helping the intramural program, the extramural program work more effectively. So that was really a different kind of job, but I really enjoyed it until last year when when a lot changed in government funding in the United States, and we were not able to do things the way that I thought they should be done. And so I decided to leave there and work for nonprofit. And I'm now working with this wonderful group, Genetic Alliance. Again, a wonderful team, amazing people around the world, clinicians and scientists, clinical labs, and genetic counselors, all working to bring genetic diagnoses to families in low and middle income countries that don't have access to genomics but need a genomic diagnosis. So that's incredible work as well. All right, I'm done.
Oh, thank you very much, Doc. That's an incredible journey, especially the way the different disciplines have come to shape your perspective. Thank you very much for that. For my next question, looking back, was there a defining moment or experience that shaped your commitment to global health and genomics?
Oh, that's a great question. In a way, I feel like there are so many. I mean, as I've just said, it wasn't where I started. Global health was not where I started. But working with H3 Africa and seeing how creative and inventive and dedicated our scientists were in terms of serving their communities well. And so, many of them were working in places where there wasn't consistent electricity, where there was, where people had to, walk a full day to get to the clinic. Just very different circumstances than anything that I had ever experienced. And yet they were using these cutting edge technologies to help people, and were able to do that was just so impressive to me. So I would say that is kind of the defining moment where it really hit home that the way things are now, the world is not fair, and where you are born makes a huge difference in what access you have, and we have to do everything we can to change that.
Thank you very much for sharing that, Doc. It's always powerful to hear how these pivotal moments shape our long-term direction. So building on from that, my next question, how has your scientific training influenced your leadership approach in large-scale programs and global initiatives?
So I think, as scientists, we're always asking what's the evidence, right? So I think that that's actually really helpful in leadership, because a lot of times decisions can feel emotional, or personal, and so if you can step back and say, all right, well, what evidence do we see that this can work? If there is no evidence right now, what evidence should we be collecting while we do this? Let's be prepared to pivot if the evidence is showing that it doesn't work, but let's try it. I think if you're leading in that way of, we're still learning, we're exploring a space, and we're exploring it together, and we're all going to reassess as we go along based on what we see, rather than we're just doing this no matter what. I think that's a helpful approach for leadership. So I think scientific training helps a lot with that. I think the other thing that it helps with is really being in science gives you a very strong, and I've used this word a lot lately, and I'll try and explain what I mean, but a very strong sense of mission, right? You know what the end goal is, you know what you're trying to get to, you know, again, a common goal is a really important thing for people to be working towards. And so then even if you kind of disagree on the approach of how to get there, you at least agree that this is what we're trying to do. And then with science, it's fairly straightforward, right? We just want to know more. We want to understand more. We want to be able to do more with the work that we've been given. We want to have the right answer. We want to have an answer that is as close to the real truth as possible. And so there is that common goal. So again, I think scientific leadership is a little different than other leadership in that there's an external measure as well as an internal measure of what you're trying to do.
Thank you very much, Doc. That's a very valuable perspective. We now move on to your scientific contributions and your insights. Your work has explored viral evolution and host pathogen interactions, particularly the lentiviruses that you mentioned and HIV. What are the most important lessons genomics has taught us about infectious diseases?
That's a great question. I mean, I think that there are sort of two ways in which genomics is very useful for infectious diseases. I mean, we saw during COVID, right, that tracking, that having that genetic information really helps with understanding how infectious disease is spreading, where it's going, where it's coming from, which gives us the tools that we need to try to stop that, right? But then the other piece is the host genetics. So the viral or pathogen genomics can be super important for understanding how it is spreading through populations, spreading through the environment. All of that is very helpful information for trying to manage it. But then there's the side of the host genomics, which is something that is much more static compared to the virus, right? It's evolving much more slowly. But if we can find variations that make a difference in the outcome with viral infections, that gives us very good information about what is happening on a molecular level, right, within the body to between the virus and host, and it gives you these areas of attack for designing treatments, interventions, vaccines, whatever. You know, how can we block that interaction from happening? Well, if the body is already finding ways to do that, that gives us insight and keys into effective interventions.
Mm-hmm. Thank you very much, Doc. That really highlights how genomics is transforming our understanding of the diseases as well as ways of counteracting them. For my next question, so from your perspective, how is genomics reshaping our understanding of disease susceptibility and resilience across different populations?
Yeah, that's a great question. I mean, I think that genomics is helping us really understand, again, not just infectious diseases, but non-communicable diseases as well. And in a way, my answer is somewhat the same for non-communicable diseases as well as communicable diseases. There are two pieces of this, right? One is just from the public health perspective, what are susceptibilities, what are things to be watching out for, what are those epidemiological concerns, essentially. So what does the health system have to be prepared for? And genomic susceptibilities, whether you're talking about polygenic risk scores or allele frequencies in various places, can give that perspective, sort of, as you said, from a population perspective. What are the places where we need to be concerned? What do we need to be worried about? Obviously, there are so many other pieces of data that need to feed into that. So genomics alone is not the answer, because it's always an at these levels interaction with environment and diet and other stressors in the environment. And so there's all that going on, but certainly genomics helps us understand one piece of that puzzle. But then there's the other piece, which is, can we use the information about which genes are involved in various diseases to understand the biology of those diseases and start to design rational drugs and treatments and preventions, because we understand more about the molecular pathways that are involved, and how those molecules are interacting with each other, and that kind of thing is actually very helpful for therapy, designing therapies.
Yeah. Yeah. Thank you very much. That's a very valuable perspective, and it's exciting and also challenging to think how quickly this field is evolving and enhancing treatments, enhancing pharmaceuticals, and life in general. So looking ahead, what emerging areas in genomics do you believe will have the greatest impact on global healthcare in the next decade?
Yeah, that's a great question. I mean, I think that many of us in this field envision a world sort of where every infant is getting their genome sequenced, and that helps us inform their lifetime of medical care in ways that we can't do now, right? So I think that that's sort of the almost magical thing that might happen in the future. But I also think that sort of the next place where we are still lacking understanding and need to understand it more is this idea of interactions, right? So whether it's with gene-gene interactions, gene-environment interactions, what a lot of things where we know there's risk, we still don't know what makes that risk come to fruition or not, right? So the interactions are a huge part of, I think, what we still need to understand. And I think so much of what's going on right now in the genomics field with spatial genomics, and then also with gene environment interactions, is sort of where this is all going to accumulate. But understanding impacts in different cell types and different stages of life and all of those things, there's still a lot to explore. So
thank you very much. That's insightful. Let's shift our focus now to global genomics and equity. In your current role at the Genetic Alliance, how do you envision advancing truly inclusive and globally representative genomics?
Yeah, that's a great question. I mean, unfortunately, the place where we are right now, as I said, it's not equitable, and it takes money to make it equitable. And that is something I think Genomic Alliance and the iHope program, which was started by Illumina and now has grown, is doing wonderful things about trying to reach more and more people. But it's one organization and it's a limited amount of funding. It's not going to be able to support the whole world, right? And so I think some of it is that funding needs to come for governments, and governments need to invest in R&D in their own countries and make sure that they are using the latest technologies in their public health systems. And certainly, you know, the high income countries are not always doing better than the low income countries either, but there is that imbalance that makes it a lot harder in some places in the world. So I think sort of what it takes is commitment on all levels. It takes commitment from the scientists and from the nonprofits and from the advocacy organizations, but also from the governments and the societies themselves, that they want to treat everybody equally and bring the best healthcare to everybody. And that's a complicated problem. I understand that in many settings, that's a very complicated problem.
Yeah, that's a very valuable perspective, Doc. Thank you very much. From your response, it shows that inclusivity is clearly central to the future of genomics.
Absolutely.
Yes. So drawing from your experience, since you've been involved in initiatives like the H3 Africa, what lessons have you learned about building equitable international research partnerships?
I think the biggest lesson is to let the people on the ground lead the way, because they're the ones that know what is true in their area and in their setting. And so it really, I think a democratic approach to science is very important. And really listening and understanding and working through the difficult issues of what things are the same everywhere, which is genomes, right? And then what things are different everywhere, which are the politics and the economics and so making sure that the ethical issues, all of those things need to come to the fore and be heard before decisions are made.
Yeah. Okay. That really puts things into perspective. And this is especially important when we consider the historical imbalances in global research.
Absolutely.
So for my next question, what do you see as the biggest barrier to genomic equity today, particularly for the African researchers and populations? And how can these barriers be addressed?
Yeah, that's a great question. I mean, again, one of the biggest barriers is just access. And some of that is financially based, and some of it is political, but just access to the things you need to do the genomics. And, there are some of the bigger biotech companies are selling in Africa, but often the shipping is so much more expensive. Often, things are just, it's harder. And so just the recognition that this is kind of the future and that everybody should be working globally and in all places in the world and not restricting their scope. But then I think another big barrier is and is going to continue to be for a while, because of this imbalance in access to funding, to technologies, to all of those things, that a huge amount of the data is very unbalanced at this point. And there's just not yet the same amount of data from people in Africa, from populations there, as there is and also, Southeast Asia, South America. There are other places in the world that also have this imbalance. And as we get then into using AI that's trained on existing data sets, I think we have to be very careful that those results are not being biased by that fact. So I think that has the potential to also increase inequities, because the AI is exploring and being trained on a limited scope of the human diversity and not all of it. And so it might miss things in looking across the entire world population. So so those are areas where I think as scientists we all have to assure that we are not letting our results and our outcomes continue that bias, but have them reduce that bias.
Thank you very much, Doc. So these are critical issues that many in our audience are actually directly navigating.
Yes.
Following from that, how can global collaborations avoid extractive research practices and instead foster long-term capacity building?
Yeah, no, that's a great question. And I think that is back to your prior question about H3 Africa and lessons learned. I think that was one of the most successful things about it was that the awards were given directly to the African institutions where the African scientists were working, where often the model is that, at least in the United States, that it's given the award is given to a United States institution, and they have a subcontract with an institution elsewhere in the world. And this kind of flipped the switch on that. And it was many of the projects had US or European or other high income collaborators, but because it was the institution and the scientist in Africa that was driving that collaboration and that had the final say on it, it was a different dynamic. And it made a difference. And so, who's driving that collaboration, who's making those decisions, I think should be, as I said, the people on the ground there. And I think that's what and I don't think that it was that the scientists ever had bad intentions. They always intended to collaborate equally, no matter where that funding was sitting. But it is a structural difference in how the work is conducted.
Yeah. Okay. Thank you very much. That's important. Let's now explore the role of policy funding and leadership. During your time at the National Human Genome Research Institute, you led extramural research operations. How can funding systems better support innovation in underrepresented regions?
Yeah, that's also a great question. And I think one of the ways in which at the National Institutes of Health, we tried to do that was to provide funding opportunities that were specifically for that. But it is hard I will say one of the hardest things to get people to buy into funding is capacity building. And yet it's so important. And it can make a huge difference. And to me, I think one of the things that funders should realize is that it has one of the biggest returns on investment, right? Because funding is always an investment in something that will grow for the future. And so if you think about it that way, it's not what I want right this minute. It's what I want for the future and what I want the world to be capable of and be able to do. A little bit of money into capacity building makes a huge difference in the long-term outcomes of the work, in this case, the research that can be done in the world for the good of all people. And so I think that that's really important to remember, is that it may be a long-term investment, but it's a really, really good one.
Yeah. Thank you very much. These are really important points because funding structures often shape what science gets done and where it's being done.
Exactly.
So following on from that, what changes are needed in global research governance to ensure fairness transparency, as well as shared benefits?
That's a great question, and I think there are a couple pieces to that. So again, I mentioned before governments. Like, I really think that and again, talking about long-term investment, so there are, the return on investment for research and development is huge for a country. And so governments need to take that into their consideration, that scientific research is actually a very valuable thing to be investing in for your country, and it brings industries, and it brings healthcare, and it's super. Again, it's one of those things where it's harder to convince people that a long-term investment is a good idea, but it's a great idea. So that's a piece of it. And then I think so that's nationally. Then I think internationally, there needs to be better coordination and more work in bringing people together. And I think there are a lot of, like, GA4GH is a grassroots organization who is doing some of this, but the talk about what are the standards, what are the policies, what are the things we can all agree on in terms of data protections, in terms of data sharing, because different countries are going to approach those things differently, and yet for everybody, all of humanity, to benefit from advances, we need to make sure that these things are open enough that everybody shares. And similarly, treatments, drugs, pipelines for accessing those things is really a global problem and not just in any one place. And so having organizations, again, the WHO, CDC Africa, there are places that do this kind of thing, but I think that there's probably room for a lot more coordination and organizations that sort of hold countries to account for how they are managing things would be helpful.
Yeah. That's such an important point, because governance is often overlooked, but it is very, very influential. Right. So as a science advocate, how do you see the role of policy in shaping public trust and participation in genomics?
Again, super important. And I think that one of the things that we have probably, as genomicists, not done as well as we could is making sure that the public is engaged and involved in the science, because I think that when they're not, there's a lot of room for misunderstanding, but also there's a lot of room, not just misunderstanding, but for misuse, right? So you have to have that balance of making sure that, and I think data sharing is one of those places that you have to think very clearly about the balance. It's very important that everybody has access, but it's also very important that everybody has data protection, right? So both those things will move the science forward. And as much as the public is involved and sees the benefits of the science, then they will have more trust in the outcomes.
Thank you very much. Thank you very much for that. That's a valuable insight. We now turn to community engagement and trust. Trust is a major challenge in genomics. So what strategies have you found most effective in engaging communities meaningfully?
Yeah, it's interesting. I've been struggling these days with the balance here in terms of not feeding too much into the narrative that people don't trust science, because I think people actually do trust science. And I think that where there is a level of mistrust, and particularly with genomics, and I think genomics is very personal, because really the genome is the blueprint of who you are as a person, right? So sometimes it can, if presented badly, create a narrative that you don't agree with. And so I think that's the danger with genomics, is that if we overstate what something means or overgeneralize, it can have negative implications for people
Yeah.
themselves.
Yeah.
Um, and also, there's just always this question, what are you doing with my data? This is my very personal data, right?
Yeah.
And
Yeah.
so I think so long as people are given truthful statements about all of that, and that so long as scientists are very clear in how they present their findings in ways in which it's not shoehorning someone or generalizing something about them that isn't true, then I think we'll be okay. I think just more conversations are really important.
Yeah. 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 Trust is indeed central to community engagement.
Yeah.
So,
So community engagement, I think, that should be the start of
mhm,
everything.
mhm,
Is those conversations at the very beginning, so everybody understands what the worries are, what that common goal is, and agrees with it before you move forward.
mhm, mhm. Yes. So, building from your response, we come to a very critical question. How can scientists better communicate complex genomic concepts to non-scientific audiences without losing nuance?
Again, I think this is something we worry a little too much about, because
Mhm,
I I actually was saying this to somebody just the other day. They're like, but it's complicated and we don't understand it. And it's like, it's not that complicated and people can understand it. They just think they don't. But if you sit down again and have a conversation with them, they do. And one of the projects in H3 Africa actually pointed out and then used, like, people that farm the land, they
mhm.
know about genomics,
Yeah.
right? For
Yes.
for humans have been doing genomics with plants and animals forever. Before we knew anything about genes, you knew about inheritance patterns and how to breed and raise, and that sometimes there were changes that you could observe in over generations that were happening. People know, in their life, because genomics impacts all areas of our life. And so if you can anchor the, okay, and what does this mean about what's going on in the cells, to something they know, people understand it.
Mm.
So I
Yeah.
think that's where it is finding what is the analogy or the place of start with something that they do understand, to then take the
Mhm.
next step to explain the thing that maybe they
Yeah.
don't understand.
Mm. Okay. Thank you very much for that valuable input, Doc. So, picking up from that point, what role do cultural context and local leadership play in successful genomic research program?
I think they play a really important role, and I think that, different social structures matter a lot. And so making sure, and again, this has to do with people on the ground being the ones that are leading and making decisions, making sure that you're engaging the stakeholders that need to be engaged in the beginning, I think is really important. And again, I think genomics is a very interesting scientific field because it has to do with the individual, but it also has to do with the community and the families, right? Because the relatedness and the genomic similarities can, a finding in one person can actually impact other people around them. So making sure that all the right people are being consulted with. And I also think, and this is something that many indigenous peoples in all over the world, in the United States, in New Zealand, in Africa, in different parts of Africa. I hate to lump Africa together as one country. It's a gazillion, right? all the different, there's indigenous peoples are always of interest in the concept of genetics and genomic history and genomic ancestry because they're the origins, right? That's the origin story. And so if you want to know about where something came from, you're looking back to the roots, and indigenous people sort of are what we have to access that are as close as possible to the roots of humanity. And so they become interesting from that perspective. And as you talked about, in the past, a lot of research has been done without participation, consent, understanding. And so a lot of indigenous people have taken ownership, rightly so, of kind of their history and their historical and scientific value, and want to be at the table when those discussions and decisions are made. And I think that's valuable and important. And it also brings up a lot of interesting questions about sort of individual autonomy versus group membership. Like, so somebody who maybe genomically is very closely related, it's part of this community, but is off in another country in another place and wants to do their genome sequencing, you can't stop them from doing that either. And so those boundaries are complicated, I guess is what I'll say. And I think, again, we will do better, and we will come up with better scientific answers if we're all working together to figure out how to navigate these things.
Yeah. No, thank you very much for that response, Doc, and for bringing out the important role the cultural context and the local leadership actually play in genomic research programs.
Yeah.
We then move to innovation and the future of genomics. So with the rapid advances in genomic technologies, how do we ensure that innovation benefits all populations, not just those in the well-resourced settings?
Yeah, and I think this, again, I keep coming back to this, maybe because I worked for the government for so long, but a lot of that is if something is funded with public dollars, you have to try to make it as affordable and widely accessible as possible. If something is funded privately by industry, they don't have to do that. And so I think the more money that governments can put into scientific research, the more likely we will to have solutions that are more available for everybody.
Thank you very much. That's highly relevant to our audiences as early career researchers. So for my next question, what does a truly global genomics ecosystem look like to you in the next 10 to 15 years?
That's a great question. Truly global. I think that what that looks like, and actually I was just at the Hugo meetings, and it was so exciting to see how many countries are starting their own genomics programs with it, to sequence in their country and make available publicly and with public dollars, genomes, right? So truly global. Again, you would have some sort of international overarching structure that was allowing everybody to participate equally and to access data equally, and that that we were thinking as a world and not just as a country or a program about getting truly global representation in our data, so that solutions that were being provided out of this information could be provided everywhere. I do think that increasingly there is, in almost every genomic program, some level of international engagement, but none of them are reaching everywhere, right? So that's the goal, right? Is to reach everywhere.
Yeah. That's very insightful. Thank you. And that vision is something many of us are working towards.
Absolutely.
Yeah. So are there any specific models or partnerships that you believe are particularly promising for the future?
I mentioned GA4GH. I think that they are working towards including more and more people from more and more countries. I think they're not there yet. But that is that engagement is, I think, very promising, both because it's a bottom up rather than a top down structure, which I think is really important. And because they really are thinking very globally and trying to be as equitable and reasonable and standardized as possible. So that's one that I think is promising. I think that there are right now going on many different efforts to create pangenomes as references. And I think that will make a huge difference. And so as those pieces that are sort of happening in all different places connect with each other, I think that will be a very promising network. I have to give a shout out to Genetic Alliance, which I'm working with right now, and the iHope program, the model being to connect clinical sites that need sequencing with labs that do sequencing, and they're trying increasingly to bring those labs and those clinical sites closer together so that it's not high income, low income, the labs in high income and the clinical sites in low income, but that there actually is a movement towards trying to get more labs supporting this in more places and more clinical sites able to access more labs. I mean, it's right now a vision for the most part, rather than actually we've gotten there yet, but that's what we're working towards. So I think that's a model that should be followed. And again, as you talked about, capacity building. So to the extent that, again, all of these are sort of drop in the bucket efforts, but they're getting us there. But H3 Africa, I think the idea that it's not just about doing the science, it's about building the capacity to do the science is really important. So those are the ones that I can put forward.
Yeah. Thank you
off
very much.
the head.
No, thank you very much for your response. Right. Now we're going to close with some advice and inspiration. And for my first question, what advice would you give to early career researchers in Africa who want to build impactful careers in genomics?
My advice, and it's whether you're in Africa or anywhere else, my my advice is always, well, I'll flip it and say, here are the best pieces of advice I ever got.
Mhm.
The first was from one of my advisors in when I was in college, but then my father actually reiterated it later, and it really struck home, which is do the thing when you're given an opportunity to do something. Think, am I ever going to have the chance to do this again? And if the answer is no, then go and do it.
Wow.
Like, just
Wow.
yes.
Wow.
So,
Okay. No,
so
that's very helpful. We actually feel it. Like, am I
yes,
going to be able to do
exactly.
this again? No,
Right?
thank you
So
very much.
don't let the opportunities pass you by. You just got
Yeah,
to take them,
yeah.
even if you're not
Wow.
sure where it's going to lead you. If
Yeah,
um,
yeah.
and then, you know, my other piece of advice is trust yourself.
Mhm.
Like, so, you know, you're in science for a reason. You're there because you have a critical mind. You're there because you have curiosity,
Yeah,
because you want to know how something works. Don't don't give that up and don't let that go. Like if you feel like you're onto something, follow it. If you think maybe somebody else is wrong and you found something new and you've discovered something that nobody has seen before, follow it. Like just go for it. And then, you know, my last one is also going to be a go for it, which is I am horrible at writing, and I hate doing it, and yet you have to.
yeah.
Um, and so this piece of advice I got from one of my postdoc advisor
Mhm.
was just like, it doesn't matter if you like it or not, just do it. Like the
Yes.
things that you have to do that you don't like, just
Yeah.
don't put them off and don't just sit down and do them because it's actually worth it in the end.
Thank you very much, Doc. That's very helpful. And I know it will be especially valuable like for our viewers and our early career researchers. My next question is, how can young scientists position themselves to contribute meaningfully to global collaborations?
I think that again, I'm going to come back to be true to yourself, right? Like
Mhm.
you if you're starting out in a collaboration and something doesn't feel right about it, or if you're uncomfortable about it, stop and think, what is making me uncomfortable? Can I fix it? Can I not fix it? So just don't let those things that your instinct is telling you, listen to them, right? Because you probably are sensing something real.
Yeah,
um, and then for again, not just for African collaborations but for all, right? I think being a global collaborator is really important. I think that it's collaborations work better when everybody in the collaboration is very clear about what the
yeah,
expectations are and what the roles are, and
yeah,
again what the common goal is, what is the purpose of the collaboration. And it's sometimes okay if different people have different reasons to be in the collaboration, so long as everybody understands that and
yeah,
works through ahead of time what is the agreement. It's actually not a bad idea. It's always a good idea to have a memorandum of understanding or
yeah,
something in
yeah,
writing.
yeah,
It's not being trusting. It's just making sure there's a shared understanding. Trust is
yeah,
important, but so is actually having things clear. between everybody so that nobody has the false expectations.
Yeah. And that is very important. Thank you very much. We appreciate those perspectives. So building on that, what continues to inspire and motivate you in your work today?
Well, people. I mean, every day I encounter people who have such amazing dedication, who are doing amazing things, who care so much about others, and who are really, really smart, and kind and wonderful. So I think people definitely keep me going. But also, there's still more for us to know. There's so much that we don't know. So that curiosity every day I wake up, what am I going to learn today?
Mm-hmm. Mm-hmm. No, thank you very much. So to close, if you could change one thing in the global genomics landscape today, what would it be and why?
I think it comes back to access. If I could change one thing, if you wanted to do genomics research, I don't care on what, on any species, on humans, on any disease, whatever it is that you wanted to do, that genomics as a tool could help you. Mm-hmm. Wherever you were, nothing would be in the way of doing that. That's what I would change.
Mm-hmm. Thank you very much for sharing such valuable insights, Doc. That brings us to the end of what has been a very engaging and insightful session. Dr. Troyer, we thank you so much for this incredibly and insightful conversation. Your reflections on global genomics, equity leadership, and the future of science have given us not only the knowledge, but also a clear sense of direction and purpose. I'm certain our audience, especially the early career science researchers across Africa, will take away both aspiration and practical insight from your words. We deeply appreciate your time, your generosity, and your continued commitment to advancing inclusive and impactful genomics worldwide. And to our audience, we thank you for being with us today. We hope this session has sparked new ideas, meaningful reflections, and a renewed commitment to shaping a more equitable future in science. Thank you and goodbye. And we look forward to seeing you again soon. Thank you, Doctor.
Thank you.